So cellular respiration is one of those topics that I hear a lot, and people describe it as very hard, having a lot of memorization, and generally just being very complicated and hard to study for. I think there's definitely a lot to memorize, but it can be simplified. There are certain things that you can realize that can help you really understand cellular respiration and how it is used and why it is used by living organisms to produce usable energy, really. So here's a general chemical equation for cellular respiration. You just have glucose, it's oxidized and it produces water and carbon dioxide. So this is just a combustion reaction, that's what it is. And a combustion reaction, in essence, is just an oxidation reaction. So this combustion reaction, if you will, is just happening in very discrete steps within ourselves, within cells, within just all life in general. It happens in a more complicated way, but in general, this is what is happening. So first, we must define what oxidation is. So oxidation is the loss of electrons in a chemical species. And this is important to understanding cellular respiration because respiration, in essence, is an oxidation reaction. So the three big assessment statements that we need to know are these three. And they deal with glycolysis, aerobic respiration, and just chemiosmosis in general. So we start with simply glycolysis. So first off, glucose is phosphorylated by two ATP molecules. This means that in the beginning, there's like this preparatory phase where two ATP are required to initiate glycolysis. So after that, you have the 6-carbon compound. It's called hexose bisphosphate. That's not important. We don't need to know that. And this splits. This is called lysis, and it splits into two 3-carbon triose phosphates. Again, you just need to know that it's a 3-carbon compound. This 3-carbon compound, or both of them, since there are two, they are oxidized, and through this, NADH plus a proton is produced. These hydrogen carriers are very important to the process of cellular respiration because they help fuel the electron transport chain, which I will talk about in a second. For now, though, just know that these 3-carbon compounds are oxidized, and afterwards they are converted to pyruvate, which is just another 3-carbon compound, producing 2 ATP. You'll notice that in the previous step, in the phosphorylation step, 2 ATP were consumed, and over here 2 ATP are produced, so meaning there's a net 0 at this point. But because this happens for both of the 3-carbon compounds, in effect, the net gain is, or the net change is 1 glucose produces 2 pyruvate molecules, 2 ATP, and 2 hydrogen carriers. So after this point, it splits into aerobic respiration and anaerobic respiration. In the presence of oxygen, aerobic respiration will occur. However, when there's no oxygen available, say with someone who is maybe a sprinter, someone who's doing some athletics, anaerobic respiration can happen in a pinch. However, this will only produce 2 ATP, and in anaerobic respiration, pyruvate acts as the final electron acceptor, accepting the hydrogen and the electrons from the hydrogen carriers. But aerobic respiration involves three important parts: the link reaction, the Krebs cycle, and then the electron transport chain. So to begin, the link reaction. So the pyruvate must be moved to the matrix because glycolysis occurs in the cytoplasm. So pyruvate must be moved to the matrix of the mitochondria for the Krebs cycle to occur. In this reaction, which moves the pyruvate to the matrix, a hydrogen carrier is produced. And note that this happens twice for every glucose molecule. The second part of the link reaction is that the pyruvate complexes with coenzyme A, forming acetyl-CoA, which is a 2-carbon compound. For those of you who take chemistry, acetyl, or this prefix, is often used as a 2-carbon prefix, and carbon dioxide. So this is important. So it's just a 3-carbon compound by way of coenzyme A is forming a 2-carbon compound and a 1-carbon compound, carbon dioxide. So then the Krebs cycle begins. The Krebs cycle begins just with this acetyl-CoA attaching to a 4-carbon compound. This is called oxaloacetate. You don't need to know that. It's just 2 plus 4 gives you a 6-carbon compound, citrate. This is the first step of the Krebs cycle, and this happens cyclically. So in the end, this 4-carbon compound will be regained. After this occurs, the 6-carbon compound is then broken back down in a series of reactions. We do not need to know the specifics about these reactions, but they break it back down into oxaloacetate, which is the 4-carbon compound, producing 2 carbon dioxide, 1 ATP, 3 NADH plus a proton, and FADH2. This happens for each glucose molecule. This happens twice. So you might notice that for each molecule of pyruvate, for each acetyl-CoA, for each pyruvate molecule, 1 carbon dioxide comes off to produce acetyl-CoA, and the acetyl-CoA, once complexed and coming back over here, produces 2 carbon dioxide molecules. If this happens twice for each glucose molecule, you might notice that there are 6 carbon dioxide being produced. And indeed, we go back to our original reaction, and we just have our glucose producing 6 carbon dioxide. So this is where the carbon is going for each molecule of glucose. However, we still need to account for the oxygen that is being put into it and the water that is coming out. And that happens in the electron transport chain. So this is where all of these NADH plus H plus and the FADH2 have come in. There have been a lot of them that have been produced in glycolysis in the KRB cycle. I believe 2 are produced in glycolysis, 2 in the link reaction, 10 NADH plus in the KRB cycle, and then, 10 in total at that point, and then FADH2. So all of these hydrogen carriers are carrying potential energy, which is as a result of them having collected hydrogen and the electrons from these things. In fact, they were reduced. So this is basically the cell's way of transferring energy from one place to another, and now these hydrogen carriers are transferring energy to the electron transport chain. So they provide electrons to the electron transport chain, and this energy is carried in between each successive protein in the transport chain, which allows hydrogen ions, just protons, to move from the matrix into the intermembrane space of the mitochondria. Note that the intermembrane space is very, very small, and the matrix is relatively large in comparison. So the concentration of these H plus ions in the intermembrane space is much higher than the concentration of H plus ions outside, since concentration is defined as the amount of things per unit volume, and the intermembrane space has much smaller volume. So this creates an electrochemical gradient. So this gradient causes passive transport, which allows the H plus ions to move back. And once these H plus ions move back to the matrix, they allow the enzyme ATP synthase, which is a membrane protein, to produce ATP. So the vast majority of the ATP produced in cellular respiration is as a result of the hydrogen carriers and the electron transport chain. In fact, 32 out of 36 ATP are produced from this electron transport chain. Finally, the hydrogen ions and the electrons in the transport chain are combined with oxygen, which is what we breathe and gain in our bodies through gas exchange, to produce water. So now we are missing 2 species from the original reaction. We have our oxygen, we have the water. So we can see that our reaction is complete. Through this, we show that glucose is oxidized and produces water and carbon dioxide. So what we need to know is just how to outline the process of glycolysis, knowing how to mention the phosphorylation, that it splits in 2, and that it produces ATP. We also need to explain aerobic respiration, up to including the link reaction, the KRB cycle, and also the electron transport chain, including the importance of the hydrogen carriers and of oxygen as the final electron acceptor, because without oxygen, aerobic respiration cannot occur. We also need to know how to explain the process of chemiosmosis, which was the process moving protons, the hydrogen ions, in and out of the intermembrane space. Thank you, and remember to like and comment on this video, and subscribe for more biology content.